Okay, so we're going to start by manipulating your data. This could be MTT, it could be crystal violet data, and this is how we need it in Excel. So you've got your 0%, fetal car serum, and some data points. You should have 12 data points, I've just typed in 8. So we've got 0% up to 20%, our 8 data points. We then need to calculate a mean, so the easiest way is to type equals, go to this box here, click on average, highlight your data points. You should have 12, I've got 8, and that's your mean of the data. We also need to calculate a standard deviation, so type equals, click on that box, go to stdev, standard deviation, highlight your 8 data points, press return, and you've calculated it for your 8 values. Rather than repeating the process 5 times and then again for your other experiment, highlight the 2 boxes, drag them across, and that will copy the function all the way across, so you don't have to just keep repeating yourself. Okay, next step, if you want to actually do some statistics on this, I'm going to show you how to do a t-test, although you don't need to do a t-test on this data, but I'm going to show you anyway. A t-test will ask the question, is this different to this? So is this column different to this column? So type equals, go to this little box here and look for t-test. If it's not there, go under more functions and search for t-test. Now this box appears, array 1 is your control data, so highlight your control data. Array 2 is your test data. Tails, let's go for a 2-tailed test. We're asking if the data is different, not specifically higher or lower. And then type, on this one, it's not a paired test because this well is not intrinsically paired to the behaviour of this well. It is an unpaired test, and we'll go type 2. So just put number 2 in that box. That's kicking out a value of 0.00682, and that is the p-value that you see on graphs. So this is effectively our p-value. And if the p-value is less than 0.05, we say that's significant, and we use this terminology. So p is less than 0.05, what we do is we put a little statistical star on the graph to denote statistical significance. So this would have at least 1 star, which is to say that the data is statistically significant. If the value comes out to be less than 0.01, we generally put 2 stars. For p less than 0.001, we would put 3 stars. So what we can see is that this data is less than 0.01, so we would say that that would have 2 stars. And on the bar chart that we're going to draw, we would put physically 2 asterisks above that bar to denote statistical significance. Now there's some simple rules in statistics, and that is you can use a t-test for one set of data against another set of data, like 0 versus 1%. But what you can't do is do 1% versus 0%, 0% versus 5, 0, 10, 0 and 20. Multiple comparisons like that are not really valid. So in which case we have to do a different test that takes into account various things that you don't need to know about, but basically it's not right to do 4 different t-tests on this data. We would typically do an Anova test or a Kruskal-Wallis test, and that's what I'm going to show you next. So what you would need to do is open up the software called StatsDirect. So if you go to the internet, type in appsanywhere.shu.ac.uk, search for stats, and it will bring up this program called StatsDirect, and this should work from any university computer. It comes up with this little box which would say update or close. For some reason you click on close, it keeps the current version of the software and it opens up StatsDirect. We then open up a new workbook, and what we see is a view that's very similar to Excel. all we're going to do is copy our data from Excel. Get rid of that. Copy our data from Excel, which is this one, and don't copy these data labels, just copy the actual values, no means, no standard deviations. So copy that into Stats Direct, dump them in column A1, and there's your data. Okay, we can do two different analyses on this. If we go to Analysis, go to Analysis of Variance, we can pick a one-way ANOVA, which is suitable for this data. And what we have to do is it tells you that something in the data is significantly different. If we do a two key comparisons, don't ask me why it's a two key comparisons, that's just what is suitable. Click on that one and click Run. It will compare A versus B, A versus D, A versus E. It will also do all the other comparisons, so E versus D would be 20 versus 10, and we don't care about that. We're only interested in 0 versus 10, 0 versus 20. And what we can see is A versus B, which is 0% versus 1%, it's coming up with a p-value of p of 0.0109. So that is, so p of 0.0109 is less than 0.05, so we give it 1 star. Now what you can see is the t-test was giving one significance value, the ANOVA is actually giving a different value. I would, in this, because you've got multiple sets of data, I would go with the ANOVA. And we'll also look at Kruskal-Wallis shortly as well. But anyway, the value that you're getting out of this is 0.0109, so you would give it 1 star on your graph. And we'll see how to put those on in a minute. A versus C, 0.01 again, so that's 1 star. It's significantly different, but not to the P of 0.01 level. A versus C, D, P of 0.0001, same with A versus E. So that's showing that there is more certainty that the 0 versus 10%, you got more certainty regarding the difference here and here than here and here. As you can see, that is roughly double that. There's also an overlap of the standard deviations, which we'll come to in a moment, but we've got more certainty that that is higher than that. Some degree of certainty that that is higher than that, but more so here. And that will make sense when you see the graphs. So on Stats Direct, we can also just go back, press return, go back to your data, and we can go and do Kruskal-Wallis instead. Now, Kruskal-Wallis deals with non-parametric data. Since we've not tested whether this data is normally distributed or not, we're going to, let's assume it's not normally distributed. We're going to pick a Kruskal-Wallis test. It does the same thing as an ANOVA, it just deals with non-parametric data. Okay, so we've clicked on that. It's this is still the old data. We're going to run our Kruskal-Wallis multiple comparisons. So if we click on Run, we've got all of these new data points. Again, it's A versus B, A versus C, and so on. It's given us significance values, P of 0.001, P of 0.0014. Again, similar to the ANOVA. So with this data, it probably doesn't matter whether you use ANOVA or Kruskal-Wallis. You might want to read up on these two tests, but it probably doesn't matter which one you use because you're effectively getting the same result out. But those data points, those significance values, are very, very similar to the ones that you got up here for your ANOVA. So I don't mind which one you use, but look up the tests and decide for yourself. So basically, you've got to draw a graph of your MTT data and your crystal violet data, two separate graphs, and annotate the significance stars for each of your data points on that graph. So I'll show you how to draw some nice pretty graphs up to my standard now. So we're back in Excel. we've got our data here. Let's say it's the crystal violet data, and I want to draw a nice graph. So highlight your means, not all of this, ignore the fact that that's highlighted. If you're highlighting those means, go to Insert, Column, and just pick the first one. And you see you've got a graph like this, showing this is going to be a 0, 1, 5, 10, 20. We want to make this all correct, so I'm going to go to Select Data, alter the x-axis labels, that's these across the bottom. Instead of saying 1, 2, 3, 4, I'm going to highlight those wells, press return, and I've now got my actual values. Get rid of that series 1, that's not needed. You can get rid of those lines as well, that's not needed. And next we need to put on some axes. So if I go to Layout, Axes, Primary, Horizontal, Title, FCS, so that's what you added. If we go to Axes and Titles, Primary, Vertical, let's have a rotated one, and that will be Absorbance 570. We need a title, so we go to Layout again, so you've got to click on the graph, go to Layout, chart title, sorry, centered or above chart is better. Simply I do the effect of x on y as determined by z, and that works for most experiments. So in here we've got the effect of fetal car serum on proliferation of PC3 cells as determined by MTT assay. It just works, so it's a good idea to use that little formula for any experiment of this type. So next we need to add some error bars, so click on a bar so it highlights all 5 of your bars. Now it should probably going to be under Layout. Somewhere you'll be able to find something that says error bars. It's different on maybe different versions of Excel. So click on that when you find your error bars tab. Do it this way. Click on More Error Bar Options. It seems a bit daft, but this is just the best way to do it. Custom, specify value, and then your positive error bar. You just highlight your standard deviations. Negative error bar. Repeat the process. Click on that little thing there, click OK, click Close, and it's drawn your means and your standard deviations, plus and minus. If you then go to, if you then click on here, you can change the colour of your bars. Personally, oops, that was the outline of the bars. If you click on the bars and click that one, you can change them to whatever colour you want. Simple rule is black looks good, any other colour looks horrendous. So let's go black. Then the other thing you want to do is move your chart so it becomes a new sheet, and that will appear as a tab just down here, just where it says sheet 1. Here, just below where the arrow is, it'll say chart 1, and that's your graph. Now, you also probably want to alter the font size because you're going to copy this into Word. Something like that would look good. You then want to insert some text boxes to type significance stars, so let's have one there. Oops, didn't mean to move that. Let's have one significant star there. Insert another one. Make it look a bit bigger, a bit prettier. Okay. And then one little tip is when you've got your graph perfect and final, go to File, Save As, and save it as a PDF. And then when you copy it into your Word file, none of these significance bars, none of these stars will move. Okay, so hopefully that will get you to the point where you've got a very pretty looking graph in Microsoft Word, and then you can put a figure legend underneath there outlining what the experiment is, and then in your report you want to describe the results that you see. Okay, just a few little tips now of how to make your graphs look pretty in Microsoft Word when you copy them in. I've just got a blank document here. I'm just going to open up a file that I've just saved, which is a PDF of my graph that I've just drawn. So it looks like this. It doesn't look perfect because things will have moved a little bit, but yours will look better than this. If you go to Edit, take a snapshot, get rid of that horrible black border, you can copy out that graph, dump it into your Word document, and you've got a very nice looking graph. Absolutely nothing wrong with that graph other than the significant stars are a bit wonky, but apart from that, it's a nice looking graph. You can then write your figure legend. And your figure legend should really be explaining in a couple of sentences what the graph is about, a brief interpretation of the result, and define what statistical test you have put. So if you've used an Anova, you would put P, let's say P less than 0.05 equals star brackets Anova. And that's like a shorthand way of telling me that this one star here means P less than 0.05 as tested by an Anova. Or if it's done by a t-test, say t-test, if it's Kruskal-Wallis, say Kruskal-Wallis. So you need that information in your figure legend. Other things you might need in there is the fact that your data is based on 12 wells, its mean, plus and minus standard deviation. Important to tell me that. You might want to explain how long it was incubated. Incubated for 4 days. So just some literally 2 or 3 lines explaining this information in the figure legend, so that whoever's reading this, i.e. me, knows that you know what you've done. And just to illustrate what happens if you copy your graph in directly from Excel, let's just take this graph. If I copy that from Excel and put it in my Word file, let's just see what it looks like compared to my PDF version. You can see the font's changed, things have gone on top of each other. Basically, it looks an amateurish mess. It's got this horrible black border around it, whereas mine is exactly in the same proportions with the same font proportions as in my original file. So that's why I save all my graphs as a PDF and copy them in, rather than copy them in from Excel, because it basically makes a mess of them. And you really don't want that to happen on your electronic submitted formal lab report. Also, if it's a PDF embedded with the text, Turnitin and Blackboard won't make a mess of it like this, whereas it might make a mess of your Excel graphs embedded in Word. So, if in doubt, put in pictures and graphs as PDFs instead of the native file types. So some last minute tips that I've been told you need to do for this assignment. You need to present data of your 3 experiments, MTT, Crystal Violet and Immuno. Immuno, you should be putting in 4 pictures, and I've put some pictures online if your experiment didn't work. You want to quantify the percent positivity in those 4 images. You should know how to do that. So identify what percent positivity in the 4 different images. You might also want to comment upon the number of cells in the flask that you actually started off in week 1, because remember, if you had 1 million cells per mil in your first flask and you had 5 mils of cells, that's 5 million cells. If you had 200,000 cells per mil in your minus fetal calcium flask and you had 5 mils, that's 1 million cells. So you can comment on the fact that you had a lot more cells in the original flask because they're all seeded at the same density 4 or 5 days before you got your hands on them. So you can comment on that. So in the lab report, you're focusing on your results and discussion. You're presenting your graphs like this. You're having a figure legend, and for each graph or experiment, you're having at least a paragraph of descriptive interpretive text. For your discussion, you're saying why everything happened. So, why do cells stain positive for Ki67? What is Ki67? What stage of cell cycle is it expressed? Comment, discuss the fact that you might have got 70% positivity. Maybe that's expected. Well, it is expected. Comment on 2 different crystal violet and MTT assays. Do they show the same thing? Do they show different things? How do those assays work? Why does MTT level go up when you've got more cells? You want to look into how those assays work and include that in discussion, and have a bibliography of reference list, all the references you've used to explain what MTT is, what crystal violet is, what Ki67 is. So basically you're explaining in your discussion why you got the results that you got. And I want everyone to use 3 sets of good data. That's why I've put crystal violet, MTT and immunodata online. Everyone got something to work, but most of you only got 1 or 2 things to work. So supplement your rubbish data with some good data from me. So I think that's probably everything you need to get this thing written up. Good luck.